Hey, everybody, this is Jason Wilson with the Curious About Cannabis Podcast. Thanks so much for tuning in once again. I am delighted today to be able to sit down with Dr. Daniel Hayden of Extractioneering, or as most of you probably know him from Extractioneering here in Oregon. Thanks so much for being willing to sit down with me and talk today for the podcast.
Thank you.
Yeah, I'm really excited to see what's going on. It's funny, you may or may not be surprised how hard it is to find knowledgeable extraction chemists or, scientists that are doing extraction that are willing to actually talk to me. There are a lot that seem very, very protective of what they do.
Yes, and I mean, there'll be some protected parts.
Of course.
And we wish we could be further along with the company. And when we are further along, we should re-engage about some of the more protected parts.
Oh, absolutely.
Today, we could talk about the science.
Yeah. As you continue to grow and evolve, definitely keep me in the loop, and we can do drop-ins and educate folks more on some of what you're doing. And to give people context, I know some people watching this and listening are probably somewhat familiar with Extractioneering already from a pretty solid Instagram following and that sort of thing. But for those that aren't so familiar or haven't seen any of your work, can you briefly describe a little bit about your academic and professional background, and then a little bit about the work that you're trying to do here?
Sure. Break it up into two parts. Of my background is I got my bachelor's degree in biotechnology at Rochester Institute of Technology in upstate New York. At the time, in 1994, that was when Flavor Savor tomatoes came out and genetically modified organisms really came to space.
Yeah.
And I really felt I was interested in natural products, but I really wanted to get into the technology. If there's somebody with a like mind who wants it to all go well, I wanted to get in and understand genetic modification. So I started to pursue that for my bachelor's, and I came in contact with cannabis at the time and the use and what it meant to me. And so I married the concept of biotechnology together with natural products like biodiesel and biomass for cardboard and for wood.
Yeah.
And I got into hemp in the late 90s. And my thoughts about how I could contribute biotechnology and hemp and specifically oil seeds, but there wasn't many opportunities as a graduate student.
Right.
You tell me your background in Mississippi. There's one other lab in Indiana or Illinois, but didn't really work with the plant itself. And so I said, well, if I just work with plants and genetic engineering and rare and unique plants, it will help when it comes to cannabis.
Right, you'll be ready.
So I went to the University of Hawaii, just cast myself far away into the islands, and worked with these plants that are weird and got these tissues that protect them from all these environments in the tropics, but we needed to investigate them. And so you couldn't just work on these things with the kits they were giving for corn and for other kind of sciences. Right. You had to make your new chemistry and to extract the nucleic acids. And really, my PhD was about extractions.
Yeah.
So they explained to you what the meaning of a PhD was, and it's creating your own piece of work to contribute to your field. And I was like, I got 70 pages on how to extract RNA from this anthurium, this really rubbery plant species. And I didn't understand it then, the context of it, but I got my publication, I got my PhD, and then I went into biofuels.
Oh, interesting.
So at the time, they're really a hot topic, and really biodiesel, but I got swallowed up into ethanol cellulosic. But that was tissue culture with wheat and switchgrass, working with the USDA, understanding how government operates and policies and procedures and health codes and environmental safety laws, etc. And so also working with really expensive instrumentation for sequencing.
And developing that comfortability with all of those kinds of technologies.
And so the tissue culture was really born there, and working with sequencing. I got through that, and it's a long verbose discussion, but into postdoc for UC Davis.
Oh, cool.
And this was on sequencing of an oil seed. So now we're getting closer to nonpolar, this oil body of a seed, and why is it acting so unique, and what are the genes involved? And so I did 3 years really investigating the millions and millions of genes that are expressed in developing oat seed because the oat kernel has a lot of oil in it.
Yeah, yeah.
And, understanding what about gene expression produced more oil. And the scientist wants to say it's this easy concept.
Right, yeah.
The enzyme that makes the oil, like the THC, the enzyme that makes the THC, you just boost it, you get more THC.
Right,
you knock it down, you get less. It's not like that
Nope.
in secondary metabolism. I've worked in lignin all my life. Secondary metabolism, that's where this pathway of cannabinoids stations itself. And so we found out that the cells that make the product of question had better metabolism. And when they were designed in the developing oat seed, when they were laid, there was more of them. So there was more of them to do the job of converting all the nutrients into the oil.
Yeah.
And that's why you have a better oil seed, not because that one last step in making the oil, there's just more of that enzyme and you have more oil. So, that concept, all those concepts in my background drove to the point of cannabis.
I was about to say, I can see how that would all come together and start to translate into a lot of the interesting things to investigate in cannabis. It's something I talk a lot about concerning cannabis. Is there's a good bit of research in a certain context that's been done, but there's still a lot of low-hanging fruit when it comes to cannabis research. A lot of things to investigate that we don't currently understand as well as things with like oats or corn, some of these like heavily researched agricultural commodities.
There was a ton of work done on cannabis in the 40s and the 50s and the 60s, mentioning molecules that to your imagination can conceive having these molecules in a larger amount and being able to interact with them. But where's been all the progress? So we celebrate a few scientists from Israel for coming up with context of crystallization of THC, but where's the work been for the past 70 years?
Right.
What else is characterize all these other rare molecules? So this kind of translates into what I wanted to discuss with you, but the people you interview are in the medical field.
Yeah.
And they're struggling to understand how to create context. And it's because cannabis is so complex, and then you don't have the right starting materials. Right. So if their starting material was a cannabis flower smoked, and then all their patients are treated the same, then I would listen to what's happening with the study. But it's just preparations, concentrated preparations, and then pills or whatever, ingestion. And so who knows what bioavailability.
Oh, yeah. I mean, there's the bioavailability issue. There's also one thing that drives me crazy in trying to decipher a lot of the clinical data that exists is the uncontrolled variables. So like you're talking about, there's if they do happen to find relatively high quality starting materials, which is relatively rare in a lot of clinical research that's gone on, it's still uncontrolled and inconsistent patient to patient. So really you're looking at individual case studies more than like a data set that can be validated and actually start to decipher wisdom, at large that would affect a more general population.
Now we're getting into the weeds, batch to batch variability.
Right.
So you prove something with one batch, and then they ask you to go back again, or somebody asks for the batch to run it in their own samples, and it's gone. It doesn't exist. They don't know how to qualify it. On a consumer level, in this marketplace, with distillate cartridges, we have the same thing.
Oh, yeah.
So what we have is broad spectrum, full spectrum. I'm going to give you some random cannabinoids, and you're the better for it.
Right, yeah.
So you can thank me later. I've created some wild cocktail, and I bestow it upon you. Heal yourself. You can thank me later. So we're really focusing on here, and we've done it. Of course, creating this marketplace, you have to create products, and everybody's buying it for the THC, so you're just going to move through. But we want to make sure that moving forward, we make sure we know what those minor cannabinoids are. And we engineer them reliably so the customer goes back and knows this product is a little bit more CBC in it, and it's for me.
Yeah, I mean, and that's something I've been trying to educate people on is, like, stop focusing so much on things like strain names, indica sativa designations, all these sorts of things, and start focusing on the actual chemistry of the product itself that you're consuming, because regardless of even the chemistry of the starting material, depending on how it's been extracted, refined, potentially manipulated in some way, that finished product, beyond the fact that strain names are unreliable and beyond the fact that chemical profiles are not strongly correlated with strain names and that sort of thing, just the way that something is processed throws a little monkey wrench into that as well. And it seems like consumers are slowly starting to understand that and are looking for that, but there's not a lot of good support for that. Once someone realizes, oh, I need to focus on the chemistry, I need to find consistent, if I find something that works, I need to find something not that has the same strain name, but has the same chemical profile. Then you get into the fact that, well, a lot of cannabis testing labs only test for 6 to 12, cannabinoids and, maybe 10 to 20 terpenes, terpenoids. They're not characterizing flavonoids. They're, not characterizing stilbenoids and sterols and, all these different things. So there's
what's the reproduction and what is substantial medically?
Right. Yeah. And so people can get sort of a false perception that even if they see a certificate of analysis on something, if it's a C of A that's coming out of, work that's being done right now, you're still not getting a good picture of what that chemical profile really is. And especially someone like you that understands, that there are hundreds, most likely thousands of compounds, in this plant.
Yeah.
That the idea of focusing on 6 to 12 or 20 is
so
limiting.
So then you focus on what is our business model? What are we trying to do? We're trying to reproduce experiences for our community. And so what is the endocannabinoid system? It's a lock and key. Your endocannabinoid system is different than mine.
Right.
If we were brothers born from the same parents, we would have different endocannabinoid systems. And each user needs to interact differently with their ratio of phytocannabinoids to provide them supplements. And we as a product company just want to make sure that when they buy their next balm or tincture or capsule or gummy or extract or cartridge, that they get a reproducible experience.
Yeah.
And so we chase that understanding, but we chase complexity as well.
Yeah.
So, and then you get merged into complexity, and how do we preserve complexity through the extraction? You said any downstream processes or how it's packaged can affect something that started great and then became something else.
Yeah. So that segues really well into something else that I wanted to make sure to talk to you about. And we can get as deep into it as you want because it might tiptoe into a little bit of proprietary stuff. But can you speak a little bit to how a lot of cannabis extracts are produced? What are some of the technologies and processes involved? How do they differ? What are the pros and cons? And what specifically in your context, what do you prefer and which ones are you a little more kind of skeptical about, that sort of thing?
So so this is the casual extract question for all extract or processing companies. They split themselves up into about a few different categories. Solvent, solventless, concentrate or extract is your first category. So I really focus on what's an extract, what's the botanical term for extraction.
Right.
So using a solvent to remove undesirables and to preserve desirables. I mean, using a solvent, people laugh about, is water a solvent in cannabis? Water is a solvent for water-soluble things.
Yeah, things that are more polar.
So, sure. So in this context, the body of the trichome is the resin. It's nonpolar, so we use nonpolar solvents. Something solventless would be mechanical, or steam or pressure, like rosin or hash or sift. And that preserves the complete resin profile, but with impurities. So it's not an extract. And then, so what's a solvent? It could be butane or CO2 or alcohol. What are you trying to extract and what are you trying to prevent from co-extraction? So alcohol has some polar qualities to it. It's going to pull other portions of the resin. There's something is not black or white, polar or non-polar. It bends how it wants to bend, but mostly in there it's non-polar. There are some water-soluble portions
Yeah,
of
yeah,
molecules.
exactly. Yeah.
So it's actually, and then you have butane. So I find butane is this miracle of God molecule, and it actually gets more polar as colder it gets. So you can actually bend it to grab a little bit more of that
You can get more
water
selective.
and where that water solubility in nonpolar is where flavor is, sort of. It's the flavors of things are in the esters.
Yeah.
And so butane is just a great buffer as well. So when we talk about science and biology, the first thing we do is talk about charge and buffers and how to prepare solutions for extraction. And so when I come up to this problem in cannabis, when I got done with academia and government and everything, it's a problem right in front of me. It's a separation problem that needs buffers. And so certain things work better than others. You have CO2. So CO2 doesn't have a hydrocarbon equivalent, although it comes from the industrial gas complex. Impurities could be added to CO2 just like
any
hydrocarbon, right? And so, but you have to bend CO2 in a few different directions. You say, I want a heavier molecule, I want a lighter molecule. In the end, I'll put them back together.
Yeah.
And so Humpty Dumpty got put back together again. It never, as I extract biology in my life and my career, I could not reassemble complexity in the reverse direction.
Yeah.
To any meaningful extent.
Yeah, I mean, that's a common issue in herbalism. I mean, going back to old roots of alchemy, and I don't want to deviate too much because we'll get to this, but this ties into the concept of full spectrum and everything. And this concept that a lot of herbalists have been chasing down for a long time is how do you capture the plant essence in basically a liquid form, essentially. And the most common way is taking all different parts of a plant, extracting it, trying to mix it back together. But to your point, when you do that and you mix things back together, you tend to find that things have changed, regardless of the ways you've tried to control them.
Yeah, cannabis presents the most complicated example of all this. If you want to extract salicylic acid from willow bark, I mean, it's right in front of you. You just remove everything and you get the one water-soluble compound. Yeah. And it brings down your inflammation. It takes your headache away. Yeah. But when you're talking about cannabis, it's incredibly complex, and oxygen and everything affects what's happening there.
Especially with terpenes that are very, very sensitive to oxidation. And
so, and then you say, what is full spectrum? And it's kind of like it changes. Don't let anyone tell you that we have different standards of what full spectrum is. I don't smoke the cannabis plant to get chlorophyll.
Right.
So I usually put that quote on your Instagram, and he says, we all have different standards. We don't have. We use cannabis for one reason, the resin of the trichome, right? And so we want that complete resin. You toss everything else out. I'll get my chlorophyll from somewhere else. I'll get my antioxidants from somewhere else. I'll get my proteins and my starch. I don't need that stuff from cannabis. I want a bioavailable cannabis product that I can use in an administrated way, which is a vapor that works best for my chemistry and evolution and species, right? So full spectrum is different for each batch of cannabis that you collect. And so and you want to carry the complexity throughout the extraction, but make it available. And so what I find throughout all my extraction and scientific knowledge is I've seen things come together and never be able to come apart again based on the chemistry you're using. And I feel a lot of that is happening in cannabis concentrates today. They polymerize with the substances on the exterior of the leaf and what's trying to be protected on the interior of a trichrome, lock it in place. And then when you go to vaporize it, almost like boiling water with salt in it, you're just getting these strands of glops flying at you. And the THC works just the same. You say like this to the product, it works, but you want all those things individual. So when they vaporize, you want them all individual, and so you can once your sensors and everything can get in there, and they can all get in your alveoli and in your bloodstream as individual molecules. And so what is full spectrum? It's taking that complexity of each batch and making the entirety of it available.
And maintaining, and when you're saying maintaining the complexity, you mean maintaining the complexity of those resins?
Yes, the resin that's in the trichome of the batch of cannabis. Yeah. So when we come to hemp, so in cannabis, we've had this structure where you get Girl Scout cookies, and you grow a whole room of it, and I extract it, and it's pretty linear. I mean, I could test a nug and the terpenes, but it's pretty linear. Now I walk out into the hemp field, and I have Jack Air, Grape Crush, Trainwreck. It's just all over the place. And they're going to try to make some product, a combination of oils, and it's just going to be like wedging a whole bunch of flavor and experience, just ramming it all together, even if it's not bioavailable, which in 95% it won't be.
Right, yeah.
It's just not going to be what we know of cannabis, which is individual selections.
And even just talking about the flavor bit, I mean, that's easy to illustrate just thinking about foods. Like, you don't want to take a bunch of different flavors of, I don't know, cakes or candies or something and merge them all together. That merged together flavor is not going to be as good as tasting each individual unique flavor.
Thanks for bringing up the food thing, because it's great. So another person's extract or technology is like an egg, flour, water, and oil mixed up and set in front of you and say, this is your cake.
Right.
And so, what we're trying to do is add a little bit of baking soda and put it in the oven and, understand what about the natural chemistry. When the first person made bread, like what did they understand about how the proteins in it had sulfur and made it connect together and made the dough elastic and made it rise? It was special about the chemistry of that. But you'd never know that if you didn't go through the correct sequence. So I think with a lot of cannabis extracts, it's like an unbaked cake.
Yeah, it's almost like
it has all the parts, but it's not.
It's like getting the box cakes.
Yeah.
Like, it seems like a lot of folks are jumping into starting labs. They're getting the equipment that they know is the basic equipment to doing basic processes, and they're making a cake technically, out of the core components. But it's nothing like if you went to a bakery and bought a cake from someone that really understands, the complexities to draw out flavors, make things rich and enticing, all these different things.
Yeah. So it's just that little bit of extra, and it is found in chemistry and understanding.
Yeah.
And so there is proprietary parts of it. So we're working a lot with the combination of CO2 and butane. And so it's not rocket science, and everybody does it actually, but we're scientists, so we just sequence it correctly. And what we found is that CO2 is the perfect co-solvent, is the term we're using. So what it does is it gets into the resin, and all the parts that would polymerize or attach or oxygen would affect the resin. We charge it with a CO2. Like you would carbonate a beverage.
Yeah.
And then we run the extraction, and with our sequence, we understand that the CO2 keeps it together, and it eschews like the butane and the solvent, and keeps its terpenes and all its complexity folded together. And then when we relax it and let it release, it unfolds and burps and effervesces, and all those CO2 kind of come off all those oxygens and those double bonded areas, and then you can really start to taste all those molecules start to kind of unravel. So in a sense, we protected all the molecules during the extraction. And at the end, there's no wax for them to lock together and to polymerize, and now you're able to pick it up and dab it, and the vapor, you're able to taste kind of each one. And we would hope each one would get into the bloodstream individually and have all the effects it's supposed to have.
And what are your thoughts about, so looping back around to the comments I was making on social media about full spectrum, what are your thoughts about producers that are issuing these super heavily refined distillate products and labeling them as full spectrum, which has become really common in the CBD industry particularly?
So we started this trend in 2016, and we brought the botanical understanding aspect of full spectrum into the conversation with our products that look like this. So these are all cured resin products. And so anyone watching might be like, this is live resin. Cured resin is a darker sap or a shatter or something. These are all cured resin products because we understand how to extract the chemistry from them. And so, sorry, I got off track there.
No, that's okay.
So what other companies are doing with full spectrum? So
Yeah.
in 2016, we said, this is not full spectrum. This is high cannabinoid because you've bred the plant so in excess of THCA that all of this crystalline stuff falls out of it. And it's supposed to be just kind of the liquid in the bulb, right? But the liquid in the bulb is terpenes every day. It's producing terpenes. It's it's a water, and it's making all these other complex molecules. And when you dry it, all those light terpenes go away, and you have mostly these cannabinoids left. And so in response to this full spectrum, everybody just used the term.
Right, yeah. It's very
because, so we called it high terpene and high cannabinoid. The liquid showed more terpenes than what was typically maybe in cannabis when you smoked it.
Right.
And the high cannabinoid had more cannabinoids than would be in flowered cannabis if you smoked it. So we said it's not that full experience, it's more in this direction or it's more in that direction.
Sort of honest.
We're not going to come and say it's full spectrum. And then they just took full spectrum and said it applies to everything across the board. All of this is the same, because if everybody keeps a level playing ground, everybody makes money and is treated fairly.
Well, exactly. And what is disappointing to me is in the marketing language used for a lot of these products, it's become sort of a meme that if you want the best therapeutic outcome, then you want full spectrum. And then there's not a solid definition of what someone means by full spectrum. And so you have, folks that are totally unfamiliar with any of this that are going to, Fred Meyer looking at CBD products and basically looking for something that has that term full spectrum on it, thinking, well, that's the best, pulling it and everything. And that's why when I was talking to all these companies, I kept getting different feedback that some people would say, oh, by full spectrum, we mean just the cannabinoid fraction, or we mean the cannabinoid terpene fraction, or some people that were saying whole plant. And then I say, are you using the roots?
Yes,
and
perfect.
And so
and they say, that's silly. No, that's silly. And you say, you just described. So, so they use it as a term of marketing. And so when I talk to you about the CO2 and the solvent that we're using, because we can't get the product to market, we can't talk about much of the stuff yet, because they're just ready to collect all that information and use it as info marketing to describe their products, because all the products are simple and all the same, 90% unknown, but they know people smoke them, they work with the THC at least. You can say, I got a product because I got high, and that's your only
benchmark.
So that's the only benchmark for all the cannabis, all the reputation that CBD has. It's how THC works on people.
Oh, well, and this marries greatly with the conversation I had with Dr. Kevin Spellman, who's a phytochemist and kind of a medicinal plant expert at large. But this was an issue he brought up that he said annoys him, is that what most people use to determine whether a cannabis product is effective or not is whether they got high off of it, and not understanding that there are all sorts of things happening in your body that you don't directly perceive in the moment. There's all sorts of changes happening.
And that's what it is. So a lot of cannabis products have the synthetic cannabinoid or the spice or some other compound or Viagra or list goes on. So even the CBD. So we look at the studies. When we were making our line of CBD, we know exactly how much CBD people need. It's like 300 to 500 milligrams twice a day. These things are saying you have 60 doses at these levels. So we feel like they're lesser. They work for some individuals who have a light tolerance and need a lesser key in their endocannabinoid, or they haven't been exposed to phytocannabinoids ever in their life. And they have that little bit of CBD, and it knocks something loose. 1 out of 7 Americans has gone to a CBD store and bought CBD. So people are asking the question, where is my relief? It's in these THC and THC CBD products is what they're looking for, and they don't have access to them. They only have access to CBD store in the South
Yep.
with hot products with a little bit of THC, right?
Or Delta 8 or whatever.
So we're all trading on THC. And so when we're vilifying THC, it really disappoints me. And so we got to reboot what is THC. Yeah. It's not a drug. It's not an intoxicant. So it's up to you and I to lead the paradigm of THC is different for you and me. It's just different.
Well, it's just all this cannabinoid science. I mean, it's going back to what you were saying earlier, but that everyone's endocannabinoid system tone is different. Everyone, their bodies interact with these compounds in different ways. And I'm glad you're touching on this sort of concept of the vilification of THC, because that was another thing I wanted to get into is going back to the CBD markets. It's taking people a while to catch on, but they're starting to catch on that, maybe these THC-free products, maybe they're not as effective as the ones that have some and the research from the 80s was showing that, if you have a little bit of THC with CBD, that it's going to, change, so
they call that broad spectrum
dynamics. Yeah.
Now it's called broad spectrum. They have a new thing to sell you. So it's a Furby thing. So the last 24 months of CBD stores are just like a fad. Everybody rushing in to see if they can be cured from these miracle products. And then everybody getting exposed to different levels and different workflows. And so you're just trading on THC and then this kind of fad of CBD. And the next one up is CBG.
Yeah, exactly.
So we'll just roll the dice and you tell you, CBD didn't work for you. Now it's CBG.
Right.
And so in 2021, it'll be THCV.
Right.
And then in 2022, CBDV. It'll be, So we'll just kind of ride down the line. And so it's a tough pill to swallow.
Yeah.
So, we get to work on the other end, the authentic cannabis cultivators. So there's awesome cultivators in Oregon right now.
Yeah.
And they are down to the street level of breeding, making all these crosses and trying to figure it out. And so they're still in the THC game, but hopefully they just have the passion for product that we'll end up kind of using in a vaporized or in an oil form that will be meaningful. So, this hemp craze, we just kind of sit back and watch.
Yeah, I mean, it's going to be really interesting to see how it develops now with the USDA rules coming out. And everyone's acting so surprised about the USDA rules. I'm like, did you read the Farm Bill?
Yeah,
I mean,
they're going to say they're coming down, right?
Yeah, I mean, the USDA regs just, they have to operate within the context of the Farm Bill. And now people are realizing that the Farm Bill maybe wasn't as awesome as they thought it was. And now they're yelling at the wrong people to change the rules, or yelling at the USDA or the FDA. And it's like, you need to talk to Congress. Because the law is set, and these are just rules and regulations to keep you in line with the law. And so
guidance.
Yeah. So the USDA and FDA don't have a lot they can really do about any of that. But yeah, it's gonna be interesting to see how the CBD market evolves and how that influences the cannabis industry broadly. And my hope is, with like a podcast like this, someone hearing our conversation right now, my hope is that regardless of what knowledge or wisdom anyone extracts from hearing us talk right now, hopefully it just at least inspires better conversations so that people are starting to think.
So we're on it. How does this get better?
Yeah.
What is it better for all the people that you interview in the past and who you'll interview forward, who are medical professionals who are trying to grapple with how do I create, make some context about this?
Right.
What are we trying to do? So on the product sense, we need to make reproducible batches and products before we start anything. And then we have to track what's going on.
Yeah.
And so we need customer feedback.
Yeah.
So we're going to do a reverse FDA study. So in a drug world, you have one drug, and you see how it interacts with the entire population. And hopefully it has its intended effects, and sometimes it just gives people boners, and the life goes on.
Yeah.
But cannabis is the opposite. It's a key. So everybody needs to figure out where their endocannabinoid system is and where their products that work best for them are, and they need to come together like that. And so we need all the different keys and all the different locks to understand what's going on. It's taking their knee pain away. It's taking their migraines away. It's giving them better gut health. It's curing their diabetes. It's giving them their hair back. I don't know, but we create the products, and we need the customers to feed back into it.
Yeah.
But their fear of cannabis and privacy laws and medical laws are all, and then the FDA and how to get away with saying what our products do is all there. But we're going to use social media and social medicine to cure this problem. We don't say this product cures cancer.
You're right.
We give you this app, and everyone else who it's cured cancer for will tell you what their experience was. And so all we had to do is curate the platform so that it works as well as it works. And then you can say, well, CBC, takes psoriasis away, no problem for everybody. And you wouldn't know that.
Yeah.
Until you make
and this relates to therapeutic applications. It also relates to adverse events too. I mean, something that has been unfortunate is due to the way the laws have been up until now, folks have not been talking to their doctors, nurses, their healthcare professionals about their cannabis use, what their experiences are, and that makes it really hard for doctors to understand what the truth or the possible risks, might be when people start talking about wanting to use some of this. I spoke to one doctor who, tried to explain to the listenership that when doctors get really reserved about cannabis and don't want to necessarily support the use, it's not necessarily because they think that something bad's going to happen. It's just that they are entering a realm of ignorance that makes them very uncomfortable, and they can't speak one way or the other about it.
They've heard bad things about drugs, and they don't connect it to this is a medicine.
Well, and not even necessarily just that. It's just like it might work, it might not. Because no one's talking to me, speaking from the doctor's perspective, because no one's talking to me about it, and I don't know what patients are experiencing, I can't speak about it one way or the other because don't know which way to go. So, yeah, this whole idea that we need to be talking more, sharing information, it's critical. And then this relates to another concept that I'm very interested in, how to measure the endocannabinoid system tone. And, we're still a ways off from measuring that in a sophisticated way,
How about like a profile, like a flight of beers, except I give you a flight of, as I come up with products in the system and they really get a good feedback and understanding, then I give you this flight to understand where your endocannabinoid system is at.
That's exactly the balance there is, it's, the one great thing about cannabis is that the safety profile of cannabis is pretty well established and understood. And so the idea that you can present a series of options, a series of THC CBD ratios, a series of terpene profiles, whatever, and people can try those out and try to understand what works. I mean,
it's not like giving an antidepressant and then having somebody commit suicide. Like it's just not the right chemistry, and you haven't been watching close enough. And it's like it's pretty safe to do these things. That didn't work for me, or kept me up all night. It didn't really cure my insomnia. It's not a life or death.
Exactly. The worst thing that might happen is you feel uncomfortable for a little while, and then you realize like, oh, maybe I took too much, or maybe that's not for me, or whatever. I definitely resonate with that a lot. Just this idea that there needs to be more education and more communication.
And so we talked about cannabinoids, right? So we didn't even talk about flavonoids, stilbenes, methyl esters, lignin amides, like stuff like reversatol and the stuff from tannins from they were supposed to cure cancer, be
drink a glass of wine every day.
The stuff what Dr. Oz keeps pitching with raspberry ketones or whatever it is. There's some evidence there, but they really can't put together studies that say this reduces breast cancer, or, but it's individual to individual, really. And what cannabis we want to be is like a lifestyle. We want you to put different types of cannabinoids in as you feel fit, like you would get vitamins, phytocannabinoids from ingestion. And so just keep on mixing them together and never let your body become accustomed to one particular thing. Well, THC, if you like to become accustomed to it, then that's fine. But these cannabinoids or flavonoids or different substances. They should just be as a bouquet.
Yeah.
And the one CBD, if you put it in your system, it's not going to work after a while. You're just hammering it in there, and your system is just going to ignore this overpopulation.
Right, exactly. I mean, that's part of, the way our molecular signaling networks are set up, is if there's too much signaling going down, then your body says, oh, we need to turn that volume down. We need to pull some of those receptors. You must not need them because
you get so much.
You've got so much. And there's been some interesting casual studies and some formal research that has shown that, if you need to break your tolerance with cannabis, just go to something that is a different cultivar, slightly different chemical profile, and regardless of the THC and CBD, those other compounds. And the buzz right now is all about terpenes, but just like we were saying that people are going to go from CBD to CBG and THCV, the same thing's going to, I feel like, end up happening with these chemical classes, that people are going from cannabinoids and now they're all about terpenes, and they're moving towards flavonoids now, and then they're going to keep rediscovering what we've already discovered about plants broadly, that these chemical classes are in all sorts of plants, and they're very interesting.
So from an extraction sense, the extraction is going away from all these complexities. So it was fun to have the terpene moment, where everybody was like, it's not just about shatter. Now I have these liquidy products, but now they're trying to make them lighter and lighter in color.
I've noticed
and that's going to be stripping out all the stuff that touches your tongue that makes it ignite is the same stuff that you can strip away. That looks like a color. And so what are our products? Our products in coloration are like beers, more or less.
Yeah, that's a good.
Come from hops. Cannabis and hops are pretty close. Yeah. So if you look at your beers, you have red, orange, yellow, and then the porter is dark, right? Yep. But we like those red, orange, and yellow colors, and you may not be able to see through them, but I could show you. We could bring out some samples because we're at our facility.
Yeah, that'd be great.
And the light going through them is different. Yeah. And so, working with Dr. Harrington out of Ohio, he nebulized this concept of spectral fingerprinting.
Gotcha. Okay.
Really simple, and the plate reader, he just shines the UV light through, how you used to analyze DNA. You see you go through the spectrum, and then it absorbs and lets pass through proteins, carbohydrates, all that. So these substances should absorb or let light pass through. Right. And so you should get a fingerprint. So you should immediately know, does this product have wax or interfering molecules in it that will interfere with my experience and bioavailability? And so maybe we can make a light that we can shine through and some sort of way to record. They have some of these devices merged with science, and of course in science you have all these devices. And then the conversion to cannabis is, can I put my brand on this and sell it to people in cannabis as a novel concept? But they all exist in science. Just in general.
I mean, that's been the racket in cannabis testing. All these technologies that are being presented as if they're brand new. And then you just call it
proprietary. I don't have any proprietary stuff. I have chemistry and my experience. Yeah. And we have, like, Coca-Cola was not going to tell you the exact sequence, but they're going to protect that flavor and that taste and everything. So maybe that's the concept. This texture and this taste and everything. We want to protect, like ketchup. Like Heinz wants to protect the bottle and the look. Right. And they don't want anybody interfering with that. It's like, make as much ketchup as you want.
Right, but ours is our own ketchup.
Yeah, but it's not ketchup's not proprietary or anything, right?
Yeah.
So when they say a proprietary blend of solvent, you're like, what are you talking about?
Right, yeah.
It's like, 25, 50, 25. That's our proprietary blend. It's a secret. So it's quite funny, but spectral fingerprinting. Could we do a device, you put the product, you shine a light through it, it tells you, even like broadcast to your phone app.
Yeah.
Is this product worth $60? Is this product worth $20? Is this product worth $5? And that's all I ask from this community.
Yeah.
Produce any products you want. Just make sure they're appropriately priced for the value that whoever's buying them is getting. So there's going to be a bunch of $1 to $5 products on our market. Yeah. But there's going to be elegant places for nice cakes and pastries and
exactly. And the whole spectrum is going to be there. Yeah, and that sort of technology, that's really interesting. That's a really cool, quick, qualitative measurement. Basically, just looking. You don't have to get super quantitative about any of the stuff.
Can you, in front of the, on the oven, there's like 4 bottles right up front, like lava cake, GMO, and everything.
Cool.
I'll show you what I'm talking about, a little bit about light. So when we do our process, how we quantify or qualitate things in science, you pass light through them, you put them on gels, you separate them, you run them through GC columns and heat them up, you bend them around magnetic things. So, you know, we started looking at the light that's coming through our products, and nobody else could show us. You could have, you know, live resin, you could have a distillate jar. They could look and appear, depending upon their heat, as our products, but you shine the light through our products, and they have a unique spectrum that comes out because they're effectively pure.
Yeah.
And they don't absorb, they don't have one of these particles that absorb that.
That's a good way to explain that. Yeah, yeah.
And so we're going to try to hitch onto that and try to, with all of our products, show how optically pure they are. And then get into the game of, well, CO2 or rosin or some of these other things. They still have wax.
Well, and that would certainly be interesting if in the future at dispensaries or wherever folks end up buying extracts in different states, but if there were a tool like that, in the same way that for flower, folks are looking at through magnifiers and, you know, smelling and everything, that with extracts, they, you know, have some mounted light or something and can compare these spectra, and then even some, you know, getting around to kind of what you're sort of pointing toward is a new kind of qualitative standard that you could actually come up with some ranking or a spectrum of qualities. Yeah. That those.
So imagine that no lab's going to do that for me.
Right.
Because there no company who can't meet that quality is going to say labs should do this test, or we should have this test mandated so that we know. Yeah, because it's not. So that's not a common theme. That's only a theme to us because we have that higher ability to reach that second tier. Yeah. So another thing is they have handheld. You put your nugget in, you get the USB reader. It tells you, and those things, they're not accurate. They give you the same result every time. So whatever thing we do, it really has to be spot on and reliable, and then that really puts the customer's trust in whatever it is. Yeah. So eventually, we want to make sure that everybody can come with the product table, but there's got to be some metric, some qualitative measure of how well you've separated the resin from this extraction, and how well you've carried it down the line, and how bioavailable it is. And bioavailable isn't if you put a nano emulsion and do all these nano and pico things to it, and you shove it in. You're just shoving it in a water soluble, and then it's disassociating in your body, and then you're calling it nano. I mean, you know, that's not what we're talking about. We're talking about every molecule being unperturbed and accessible under your administration regimen.
And you don't necessarily want 100% of cannabinoids and terpenes and all these other things to be bioavailable. I mean, we've co-evolved with the idea that, you know, a lot of cannabinoids we ingest do just get excreted, and that's fine because it doesn't take much to, you know, get in your bloodstream, reach sites of actions, and cause profound changes to the body. And so it's something that that actually, there are all sorts of things about the industry that make me a little nervous. It's not that I necessarily think there's going to be some sort of crisis or anything, but I'm keeping an eye on it. But attempts to change bioavailability and things like that, it's like we just don't really know what some of those effects are going to be.
So the THCO acetate, I said was going to be the first thing that killed somebody in cannabis. So people were making this molecule, and it's 100 times the efficacy of THC, which I don't know. You're having a problem with the efficacy of THC?
You gotta.
Okay, so what happens is it gets in the same stream. Somebody does the amount of product they would use custom with THC with this product that's 100 times more, and they crash themselves out. And then you have your first death in THC because everybody's cannabis is so safe. How could we do any wrong? And then you add the O-acetate to it, and now you're doing wrong. So anytime I see it come up, oh, we're doing THCO acetate. This is a new thing. I try to squash it immediately and just be like, you're going to kill people.
Well, we've
Don't
already been
do
through
it.
this with synthetic cannabinoids.
Yeah,
I
like-
mean, we've dealt with these ultra potent cannabinoids and seen what they do and make people pass out, throw up immediately. People crack their skulls. People that, especially, so, we mentioned earlier off camera that I'm from Mississippi, something that was really common and is less common now, but in Mississippi, where prohibition is still so strict, no one wants to fail a drug test, but people still want to use cannabis, and there's not always necessarily a sophisticated understanding of how quote unquote legal
like
bud
a
or
nug
spice or
or
K2,
or spice.
how that's different than
Yeah.
cannabis. And so a lot of people are using, these herbs with synthetic cannabinoids sprayed on them.
Comes up in athletes
Yeah,
occasionally.
yeah,
like
it's
you hear
very
somebody
common
showing
in-
up at the police station all whacked out.
It's very common
they
in
wanna
high
fail it.
schools with athletes that don't want to fail drug tests and stuff. And we already know that there are major problems with those things, that ER visits are, that's something that doctors do have experience with, particularly in southeastern states and in the Midwest and stuff, where that's exactly, yeah. And so absolutely, I'm with you there, that we've got to be really careful about introducing
There's nothing wrong with the amount of cannabinoids that's in cannabis. Let's just start getting them
Right.
bred up, right?
Right.
Start to increase
make
the
high-quality
levels
products and-
of those minor cannabinoids.
Right.
Through breeding, we can get them all back. The cannabis plant is like a treasure chest.
Yeah,
If the biopharma found this in the Amazon today, it'd be the investigation of our entire lifetime.
Yeah.
We found this miracle drug plant.
It's a
but-
biochemical factory. It's amazing.
So they're all in there. So we just got to start teasing them out. That's what my upsetness is. So you characterized THC in 1950, and here we are just sitting with THC and a little bit of CBD. And there's
half a century.
everybody wants to
more
say
than half a century.
how
later.
many cannabinoids are in there? 35, 104. Different context, bonds changing.
Oh, exactly,
just
yeah.
ornamentation, stereochemistry. But they're all in there. We just need to breed them out.
And
So we'll be ready when it comes to making the high quality vaporization products from any new breed with any cannabis elevated.
Okay, cool. So we're back from a very brief break. So in case those of you that are watching or listening, in case we didn't mention it before, we're actually recording this podcast episode in a licensed cannabis facility here. And so one of the benefits of that is we actually get to take a look at some of the products that we've been talking about up until now. Can you just kind of...
So this is kind of bulk products. So
Okay.
an individual customer who gets a gram from the dispensary or gets shown one would never encounter anything like this. So it's important to know in your products and your consistencies what makes this individual gram, what it looks like in its bulk form. And so we want to make this process transparent when we get going with the customer. We want to show the plant as it's growing. We want to show the material and have it tested ourselves and understand the terpene profiles of the material used for this. We're not afraid to show you trim and show you nug. It's all the same stuff to us. If you're shopping for only nug run, you can go through our catalog and choose them wisely. But
Nice,
to
okay.
us, cannabis is cannabis, trichome is trichome, the resin in it is just as good. So this is what our products look like in bulk. And so here are the outcomes of our extraction, more or less. And so to show you the uniqueness of the extraction, this is the wax in the extra THCA that we take out of our product, and this is what we leave behind. Now, this is only a portion of it. I probably have a jar like this of the liquid terpenes, and then the extra THCA, but this has all of the waxes that would polymerize and lock all this together removed, and they're all in this product right here. Now, we have to say, well, this is the non-winterized. Well, this product is like rosin or hash
Okay.
in its flavor and consistency and in its profile. So it's not like this is the undesirable from this.
Gotcha.
But that's the conversation. But this product is just a lesser in cost for the customer product because it's not as refined as this. People absolutely love this product because it's cannabinoid ratio. So whenever we do the extraction, this cannabinoid ratio is at a specific amount, and it collects more of the heavier cannabinoid molecules, and it's a more deep, dead set high. It's too much for me. So it'd be like indica, sativa, if you want to go that way, right?
Right.
This product is for the daytime, the luxury use, the flavor use, especially the liquids. And I like, and then the sugars for a little bit more potency when you do it. But because most of it's THCA, the liquid is THCA and terpenes, it's at that right balance where you can just get all of the flavor. And when we have products in bulk like this improves with age. So it's naturally gassing off. So
I heard it.
Yeah, it's going to gas off. Try. Yeah, there you go. So there, we took them out of the heat. So there's a little bit. So it will naturally gas off over time, and the flavonoids will separate, and they'll be a little bit heavier. And so don't tip them over yet because I think I got them all cracked. But as they keep them in there longer, the heavier molecules in suspension will naturally fall down.
Right.
And you can have this vintage product even with flavonoids that is the most wonderful product I've ever tasted, and you can't step away from it, which would be you leave this sitting here for 6 months and you pour off most of it, and what is down here you dab, and it's the 100x flavor, just all the flavor and everything.
Wow.
So these different fractions have to be scientifically analyzed, and they're with $1 million machines that I can't get access to banking for. So but knowing they exist is enough for us. And then going through the different versions of the extractions, again, all these are cured resin. And so this is the wax that was taken out of these. And so that is a whole product lineup for all different types, whether you want value and more potency or you want more flavor and a little bit lighter. And so kind of that conversation, we'll place indica, sativa in our lineup.
Yeah, that's reassuring because the indica sativa thing drives me crazy because it's largely devoid of meaning.
And so now we were talking about the spectral fingerprint of these products that don't have the wax. Oops, sorry. So the wax, let's see if I can get them closer to me. Wax is taken out. And so you can see the light, and up here is from your angle is what you're looking at. If you held a paper, you would see a ring of a specific color, and that's what you're talking about kind of the spectral fingerprint. So how the distance that the light passes through and then the spectrum that it is would create a quality of extracts. And so in this cluster is one product that didn't winterize as well as we would have liked. So we can see, but these are darker products. They still have that glow, and these are all different batches and different extractions. You could still see that one glow. And this one didn't winterize as effectively as we would have liked it to.
Absorbing
so all those molecules that we wanted to push into here for this batch, we kind of failed. And so in that context, we obviously optimize processes to never let that happen. But it would go into a different product lineup. So we create different product classes where all our successes meet certain grades, and then our in-betweens, it didn't work because of whatever reason, but it's still in a better quality class, goes in the middle, and then products that have the waxy fraction that are more natural, sedative products are in the accessible price range.
Yeah, that's super interesting. And then within each of these classes, you can have such a spectra of, to an extent, of chemistry with those two to tease out different effects or different options for people that prefer a product class. but then are looking for some variability within that product class.
So the complexity we would want to do is these are your full spectrum natural preparations, and then we could add cannabinoids based on the user to these natural flavor preparations. And so if we could create a device that would in one vial have this, and then in other vials have CBD, CBC, THCV, and then you could program, in the morning I want the haze flavor, but I want CBC and CBD.
You can make your own mix.
And then later in the afternoon, I want to put the THC in and get to the perps. So, that we can do in combination, but still we could still have isolations and complexity together, and the user can kind of determine what way they want to go. So we want to make it like as user friendly as possible.
And are these products going to be, I think you mentioned before, but I just want to clarify, are they going to be vape pen friendly?
Yes, all of our products since our inception have really driven towards the liquidity of the product. We feel that in the trichome, it's liquid. And so that's how the resin wants to set itself up. And so when we run these extractions, we want to see the liquid result at the end, and we want it to be balanced. So it doesn't, see people say myrcene is toxic at a certain temperature. Well, no, this is a complex preparation. It's in balance. And so when you're putting it in there, the myrcene in there is in this concert of molecules, and it's all, and we want it to taste well and to have an appropriate effect. So we're not going to overdrive the terpenes here. We're going to keep them at a level that the natural extract wants to have. And so with THCA, it works well because it will stay in suspension at a certain level, which I wouldn't know because the lab that tests for it. Well, I know where it is in SC labs. But if I give it to another lab, so I'll give it to another lab and they'll say it's 33% THCA. SC labs will say it's 56% THCA. Both of them will have 20, and then I will say 25% terpenes, and they'll just be 25% unknown. And that's because the chemistry with the CO2, they have to do a whole new SOP. You
got to validate for a unique, there may be a compound that is essentially THCA, but maybe has some slight ornamentation to it or something that will eventually break off or whatever, but they can't technically call that THCA.
I don't know where the problem, so I want to know what the other components are. So I can say how many terpenes and how many cannabinoids are in here. That's interesting, but how much is of the remainder? Truly. And no lab can tell me that. Only I can buy a $560,000 machine and pass these products through and say, there are my terpenes, there are my cannabinoids, and then the minus is the rest.
The great mystery. Well, just give me about $5 million.
Well,
I mean, just get $500,000 for an LC triple quad, and
that's the experience you had with the licensing. If they were professionals and asked for your advice and solicited all the angles that you knew was the appropriate way to test for cannabis, you would still be in that industry and you would be flourishing, and we would be the better for it. But
well, I mean, it's something that when I was working in labs, I mean, my whole angle that I always was interested in was research. And unfortunately, the way that things are structured, investigative research is really not incentivized. And it's for a number of reasons. But exactly, and it's one reason why I'm working with some university labs now and helping them understand cannabis and kind of go through that initial learning curve, because that's always been my interest, because there are labs all over the place that are commercial labs that will characterize stuff, but there's a lot of method development that needs to be done to get producers and consumers the information they need, and it's just going to take a long time to get to where I'd like to see it, but
that's we want to validate this stuff. I want to give this to the next person you talk to and see. We have a lemon remedy is one. It's a 3-to-1. It has a limonene, but it has some of the OG in it. He bred it really carefully. I mean, people who have epilepsy swear by lemon remedy in particular and the extracts that we produce. Also, we have, I don't know how to say the cancer of the sinuses or whatever, but somebody who used that as a vape cart, and it cured their morning illnesses and their pain and everything with it. So if I could give them like liters of that's bioavailable and ready to go, and I could say, do whatever you want, capsule, vapor pen, topical, but you have like a 5-gallon bucket, and then you only need 300 to 500 for each one. So that's the level we want to get to, is to provide, but we got to go through our understanding of what makes this now medicine, because it's not a single molecule.
That's
so why is now, what's the meaning of this? And it's in a spectral fingerprint, and it's in the molecular characterization, but then you first have to understand it. So I had to run all 50 of these things through to ask the questions first.
Right.
I don't even understand what question to ask until
that's a wonderful point. Is where we're at is trying to understand what questions do we need to be asking so that we understand what systems to develop to start to generate information to try to get at answers. And it's a super slow process. And something that popped in my mind thinking about this that I wanted to get your opinion on, preparing extracts like this for vape pens, do you see any reason that a producer would need to add non-cannabis derived additives to a product to make it suitable for a vape pen?
Yes. So in the same arc as any food company who puts sugar and flour together would call on the industry to provide them tangerine or coffee cake or whatever flavor that they need. So the neutral preparation of THC, which is the big molecule that everybody's in love with, like alcohol, like Absolute vodka. Absolute vodka is all those spirits, all of those aperitifs, and all of those things that they add to cocktails. Those are all botanical preparations as well. Those are drank with alcohol and into the sinuses and into the membranes. So we do ask the food industry to come up with terpene preparations. And when I went through this whole vetting and verification process, I looked at the companies, I looked at what they did, how they represented themselves, I looked at their product lineup, and I started to move away from preparations that had VG or PG or any separation in the flavorings, and only things that were completely stayed in suspension and were terpenes or esters that the food chemist knew that they were going to all be in homogeny, basically. And I knew that when I add that to my THC distillate preparation, that it would blend and not separate, and that it would create the experience that we discovered from these terpene products. These terpene products are at 20 and 30% and a certain cannabinoid value. And we put them through all different temperatures and devices, and they responded well. So we make our preparations in a similar vein, but we make sure that they blend well and that they are from biology, from nature, in a similar polarity. They work well together. They're not vitamins or proteins or
or hyper concentrated. I mean, one thing that you sort of just touched on is like there's got to be some kind of limit as far as when you're adding, for instance, in that context, if you're adding terpenes to a distillate or something, I imagine there's some upper limit. This is great.
Catch yourself in the thought. Catch yourself in the thought. Catch yourself in the thought where I can't just allow people to haphazardly add as many terpenes as they want and give them to the public. But on the other end, the person doing that has to be accountable. They run a company that's licensed in this state. And so, so we have different practices that are based on our experiences, and we use all of these products, and the first line we went through was our friends and family, the people we trusted most, and then we told them to bring their friends and family in. So this is our FDA trial. All of our loved ones, the ones closest to us, who had to protect us, and they, did all these products, and they came back, and every single last time said, please, can I have some more of that? Or in one or two cases would be like, we didn't have a good experience with that flavor. I remember I went in the direction of mint. People like mint and menthol one time. And that is as a food scientist, very touchy, very strong flavor. And so they just had a negative reaction. But then there's the 2 or 3 that were like, where's that mint card? I love that one. So this is what it teaches you about your customers. For every 9 you upset, you're going to have one who's in love with it. And you try to make 9 happy and have the one person who has the bad experience and try to deal with them. But it's just being accountable to who uses the products. And so you say that a legal entity or a lab should tell me you can't put more than 18% terpenes in that product. You're going to hurt somebody. And I said, I routinely put 25% in. And I've sold 25,000 cartridges. So with no feedback or no problems whatsoever. Effects, that's the danger you catch yourself in. Because I do agree. I want to stop people from doing things. I want to stop people from selling CBD 300 milligrams for $79.99 on the internet. You know, they don't deserve. People who earn that money don't deserve that because they're trying to get cannabis. But I really want to say you support legacy brands, support people who've been in the marketplace for a long time and are struggling to be legit. And those companies have to support their customers, and they all got to kind of support each other, not just I'll get it from Bob, I'll get it from Joe.
Right.
Because it's on this, it's good, this is bad, this is whatever. So.
Well, and it gets into this idea that we talked about a little bit before, that, not all extracts are the same, and, you kind of have to be really discerning about the companies and the business practices and everything, make sure they're actually putting in the care to evaluate how products are affecting people before they push them to market and that sort of thing, and what their basic business management practices are. One concept that came up in one discussion I was having with an herbal scientist friend of mine was he was expressing his concerns regarding quality control and extractions, and how he's aware that some folks that are producing extracts that don't have prior experience, prior lab experience and that sort of thing, are using hoses and containers and things that are not chemically compatible with high terpene content things. And it was something that my friend was familiar with because he's worked with essential oils a lot with other plants. So he understands, you've got to know your chemical compatibility and what's touching what might be leaching in. But not all companies necessarily have caught on to that necessarily.
It's huge. I think one of the questions here we'll go into is, what are the misconceptions about our cannabis extraction technology? And that's one of them, butane. Butane, the vilification of THC, and the vilification of butane. So if I own a CO2 device or that kind of workflow, I call butane toxic and dangerous, is the 2 words. It's dangerous, meaning if I use it in my garage and my pilot light blows myself up, I guess it is dangerous. Butane is a very stable product. You couldn't tell the difference between butane and gasoline, kind of, if I put it in front of you. And it's a very stable solvent and a very reliable.
And commonly used.
Commonly used in other industries, but also classed in this LPG category, which is propane gas, propane fuel. So with the fire code and with building code, so with science and industry, you get classed your building rated Industry 1, and you just do kind of whatever you want to bring in there. You have professional architects and professional fire protection people, and you've done it before. You're following the same practices that have been done over and over and over again. Nobody bats an eye. But as a cannabis industry, you have to rewrite the wheel for fire code and everything.
That's what it seems like,
So it's not toxic and dangerous. And the second thing is it's biodegradable.
Yeah.
So, it's the number one gasoline car emission, and it doesn't contribute to greenhouse gases. Butane and propane are simple chain molecules in the atmosphere. They coalesce and fall back down to the earth, and they're used as food for microorganisms.
Yeah.
So this is the chain of a simple hydrocarbon. My understanding is they don't want us to do that. They want us to recover it. The only trend there, I'm not going to reuse it in processes because you ask any other processes if they reuse a raw material that they can't essentially purify, it would affect downstream processes. So you can maybe you can burn it in like a propane. So, but I'm creating CO2 now.
Yeah.
So our dream workflow would be to procure hydrocarbons, pure hydrocarbons, process using once, burn them to generate electricity, catch the CO2 to make dry ice,
and then use that as a co-solvent again.
And then roll this process over again, but that comes with resources.
Right, yeah, no, I definitely like that idea, that sort of closed loop thinking, but beyond the stainless steel, like the simple, what people think of closed loop systems, but closed loop
just a closed, but then and then it's right with what it comes in contact with. We only come in contact with steel, glass, PTFE, silicone, and that's about it. Ceramic is, I think, the last thing we come in contact with. And all those substances are inert. The PTFE is incredibly important. Because it's an incredibly inert compound. It lines all these caps.
Yep, exactly.
We don't recover stuff that touches certain plastics. Only the PTFE is against.
That was really big in all of my lab work. Any holding samples has to be PTFE lined lids and things.
And as far as the silicone is concerned, it just absorbs. It doesn't leach or contribute any sort of negativity. That's the concept of silicone. Any sort of other plastic or polymer, hosing or gasket could leach in the other direction, depending upon how it was made.
Yeah, and sometimes you can taste it.
Yeah, BPA. So there you go. And the last thing about what I just talked about, it applies to cartridges as well. So it would never put any of these products in a plastic cartridge, unless it was PTFE. So you tell me that would probably be a $15 cartridge pod.
Right,
if
just
the whole
for
structure
the
was PTFE. Yeah. So what I found out about the heavy metals in cartridges, some of it's the casting, it's the molds. They pour all these molds to make all the casts, and they're reused over and over and over and over again. And so they just have to pour a virgin mold, which is how they solve the heavy metal problem. But most of them come from black plastics. So the black plastic tip is where a lot of the heavy metals are detected, because that's where the cheapest molds are used to make those tips.
Interesting, yeah.
And then you add a little bit of terpenes, and you're getting some leaching. So it just draws a parallel. Well, there's a ton of cheap Chinese plastic everywhere in our life and in all of our goods and surfaces and whatever we buy and all of our new stuff. So we come in contact with all of that, all those toxins all the time. I'm not trying to say this is safe.
Right.
I'm trying to explain
just the reality of our situation.
Toxicological thresholds in life as we know it.
Well, and the dose makes the poison, right?
Yes. So we keep with all ceramics and all glass and all steel, and we know those products after using them over and over again and putting these things in them and letting them sit for years in back seats of cars and under wherever and pulling them back out and knowing there's no degradation or etching or anything like that. No off taste even after that long period of time. So we're confident keeping to those substances are good, but like a pod, we don't put our products in pod because we taste that tip right when we break it out of its package, and we taste that plastic tip from interacting with the terpenes. So it's not an authentic experience, and it's easy to eliminate.
Yeah, and it's something I hope all of that gets more attention, because going back to talking about the safety profile of cannabis and everything. Cannabis is a pretty safe plant to interact with, and even extracts seem to be very safe. And it's the contaminant piece that starts to cause problems. And what I've been trying to wrap my mind around lately is the whole quote unquote vaping crisis and what's going on with that, since it tends to affect primarily black market vape pens, and primarily cannabis vape pens, although there's some of it's related to nicotine as well. And trying to understand what's going on with that. Is that cheap cartridges and things are leaching in and people are getting sick off of that? Or is it additives? And no one's been able to identify a common thread, and there may not be a common thread. There could be multiple things happening simultaneously.
Okay.
But
2 scientists sitting in a room.
yeah.
Let's figure it out.
Right. Let's armchair science. Let's go.
So it didn't happen in 2016, right? So like we could just list all of the things that happened in 2016.
Yeah.
Pesticide contaminated vape carts, bad hardware, cadmium solder, all these things that have been speculated. People using vape carts all the way to the end, too high temperature,
right.
Which I'm going to explain what the situation is now. All of those things have continually persisted in cannabis. Cannabis with neem sprayed all over it, with bugs all over it, mildew.
Myclobutyl's been around
We
forever.
know the individual examples of oversensitive individuals who have hypermucus and who have sucked in mold, and their lungs will never be the same, which I communicate with them on Instagram.
Yeah.
For them, it was different.
Yeah.
They ran into a system, the right combination, lock and key happened, and for them, but everything else was fine. You could put olive oil in a cartridge, and there was nobody really dying. So there was a event. And so it's like a virus event. Where is ground zero? Where's the monkey running around in the forest?
Yeah.
So it is a batch of something that was produced in China. It could have a batch of that something could be made every week, every day, every 2 months. It's a specific volume.
Right.
That batch of whatever it is now used as an ingredient in a third thing. And that third thing gets ingredients from all different places, but didn't have a batch that's substantial here. And that batch of whatever that ingredient was is used in 100 things.
Yeah.
Now you have the human element. So now you have to have somebody put it at a certain amount in a certain level of a vape cart, 25, 35, 45%. And then this last element is hardware and psychology. So my thought is, after all the chain of custody I just explained to you, what hurts the problem the most is people every day could smoke that and never have a problem.
Right.
Only under a certain circumstance somebody dies.
Yeah.
And when that happens, I think it's somebody using a cartridge all the way down to the end, the nicotine mods, putting that too high and getting them. But you could do that with every other cannabis preparation, VG, PG, and not hurt yourself, all the same thing, but with this one particular batch that came from China that has like a vitamin E acetate, but with a certain ornamentation of impurity that...
Well, and it does seem, the latest report I saw from the CDC said that it does seem like the cases are plateauing and starting to go back down, which then begs this question, if it was one or a handful of batches of something that folks were using that had a unique contaminant in it or whatever, maybe that's already self-resolved itself. We don't know. We have no idea.
Yes. But everybody wants to know.
Right.
So, and I want to know.
And people want answers.
And you would feel like, I know where all my cartridges are that I've used in the past 3 weeks.
Yeah.
And so I don't understand why all the cartridges are right in front of them. Yeah. So why they can't reproduce what's common among them is frustrating. And the paradigm in the media that is adding fuel to the fire of it's THC that's killing people is bizarre. That's
been a very interesting thing to see, that headlines are like, it's THC, and I'm like, no, I think what you're trying to say is that it's common in cannabis products, but that's because between nicotine and cannabis products, just in what we've been describing, cannabis resins and things, it's different than making it nicotine e-juice. and requires different types of preparations to make them suitable for vape pens. And so it makes sense of why you might see something that affects cannabis vape pens more so than nicotine, because they're very different products, even though people often talk about them as if they're the same thing, that it's all vape pens, it's all vape juice and stuff. And it's like, no, we're talking about very different products handled different ways. And
yeah, but like I say, there's that one workflow, like you
Right.
vape nicotine, you add a thickener.
Yeah.
THC, you add a dilutant.
Right, exactly.
They're both needing flavors. So like, where the blend was, or if they used something to flavor that had the same stuff you'd use to dilute, that's where your cross up is. And then the last piece about it is somebody can take my package and put a cartridge in it, and somebody could die from it. And because of how this industry is on the media and everything, you never know. It wasn't our product. It was just in our packaging.
Oh, exactly. Yeah.
And that is really disappointing to see from happen to certain brands,
Yeah. I've seen several that it seems like black market groups and companies are straight up using common brand names that are in the legal industries. Just obviously super confusing to people. And
it's the funny thing is the rat race trying to monetize how to take care of that. So you're trying to offer sticker people buy to authenticate. You just got to go to a dispensary and buy from. So how we don't need a sticker on our packaging to authenticate it. We're just going to show you exactly where all these things came from and who the vendors are we're dealing with.
Exactly.
We're getting down to the QR code on each piece.
Yeah.
So the customer will ping the piece and it will show you where it was bought from, what dispensary, and then it'll show you all this batch information and everything about it. So and then it'll show you only 133 of these pieces were created. You got number 59.
Right.
And so,
well, and this all relates to, the work that I do in quality management consulting. Like, basic good manufacturing practices can help deal with a lot of this, because anyone that's producing anything that someone is consuming should be vetting their suppliers, should be understanding the quality of the materials that they're using, should value third-party testing for things that aren't necessarily required from a regulatory perspective, but are important for you as a producer to understand the quality of what you're making and what consumers are interacting with. And at the end of the day, I view issues like this that are related to quality as an opportunity for producers that are doing things better and are doing things right to step up and market that and say, like, this is what we do. While there's some proprietary stuff, like, overall, we're kind of an open door as far as, being able to demonstrate, just how safe we're trying to make these things and what we know, what we don't know. And
because we're graduating into the legality of it.
Yeah.
Because we're being licensed and authorized to license directly to customer, because we can take our face now.
Yeah.
Instead of hide behind a brand or a Ziploc bag or a logo.
Right.
We can be more transparent. And so that's what I think is a great marketing tool. And even things like these bottles. So these bottles come from an environmental bottle company, so they're free of any pesticides or heavy metals. They're tested internally themselves, and they're provided to people who do environmental testing.
They remind me of water collection.
They're
bottles.
water collection bottles. Yeah. Yep. So my products are being graded on that metric, and so I'll just get my bottles from somebody in there. They go and they clean each one by hand, and then certify those whole lots of bottles for that industry. So they're the same price.
Yeah.
So in my workflow, why not get them from that manufacturer rather than somebody who got them from China and is cannabusinessbottlingcompany.com.
Right. Yeah.
So that's the choices that a scientist makes. And then when we were talking about scientists, about vetting your own things and making, as a scientist, I fail every day. We fail every day.
And that's part of the process.
Those are our most important attributes, is understanding why we failed and how we improve.
Yep.
And we're not trying to never fail, because it's not teaching us something. We're always trying to improve and understand that kind of sequence. So when we make these cartridges and these products, we're looking, very over scrutinizing those points, and we want to be successful. So
and hopefully that mentality spreads throughout the industry.
So I mean, yeah. So how we're going to give that approach? We're going to put products on the shelf that have that full transparency, but we have to understand our products.
Yeah.
So we have certain products that we will be debuting in videos again where these products all bubble.
Oh yeah.
This bubble is attributed to carbon dioxide.
Mm-hmm.
So we've bought devices to evaluate carbon dioxide being released.
Yeah.
And so you know that it's not butane.
Right. Yeah.
On public media, when you can just see, they're going to accuse these products of being high in residuals and full of butane because they're bubbling. And so we have to understand marketing and how do we present this to the customer so they can be assured that this is carbon dioxide like a soft drink.
Right.
And then all of these are residual free. Along with that comes mandated testing that they ask us to do, and that will all be passed. And so we can contribute that with every batch and then have them transparent. So people start to say, I'll look for this consistency, this style, this type of product, because I know that they have all those good practices.
Yeah.
So it's really but it's been a 6-year march.
Yeah, no, it has. And on the topic of the lab testing, the third-party testing, what are your thoughts on that for extracts right now? Do you think it's adequate?
So let's start with what lab testing is necessary for. So in the beginning, 96 cancer patients had compromised immune systems, and you wanted to make sure if they were going to ingest something by their lungs that it was free of mold and bacteria, and it's dangerous to AIDS patients. That's what the purpose of medical cannabis is for.
Yeah.
These end-of-life candidates. So that's kind of where the lab testing sprouted from for the cannabis aspect. But, that is not a reliable measure because over time, mold and everything can grow just over time. Right. Not at that one moment in time. Potency is a complete throw-off as well. So it can be gamed. Taking one sample of one garden is the most ridiculous premise. You got to pay $130 to get your product on the market to have an entertainment test. It should be like 9 replicates of 9 in a grid. If you want a true kind of concept of potency. And so we deal with it every time we get batches of cannabis, and it gives us a result. And so we do things reproducibly over and over and over again, and we know exactly what the potency of cannabis is, and it's nowhere near in the middle of the 20s,
Oh yeah, no, and it's something that when I was working with the testing labs, we get questions about this all the time of like why, particularly in infused product manufacturers, people making edibles or topicals, and like why are my potencies coming out inconsistent? And we try to backtrack, and it's like, well, what are you using as your inputs? What testing are you doing in process and everything? And more often than not, what we would learn is that they were relying on the test result of a flower and then doing math based on that to figure out what their target potency is. And they weren't even really doing in-process testing on like the infused butter or olive oil or whatever. And so the first thing I'd have to explain is, well, you realize that that potency result on that flower is a snapshot of that very specific bit of plant material.
But even people think it's 1 gram. I mean, you're talking about like 100 milligrams.
Yeah.
It's like a calyx in like a fan leaf.
Yeah, it's usually 100 milligrams to 500 milligrams. If you're assuming that the potency is going to be low, maybe it's a full gram. In hemp testing, that's often the case. And of course, there are ways to adjust that. But yeah, I mean, you're taking. One thing is now they adjusted the sampling a couple years ago, so now you've got to take, the 0.5% of the lot, collect that up. Theoretically, you should be homogenizing all of that material together. However, 1, there's no mandate for labs to do that, so they're not all doing that. 2, even when you try to powder all of that material together, you still deal with sampling biases and some heterogeneity still in that material. And that's a hard pill for some people to swallow, just how heterogeneous a natural product is, which once again, coming from a natural products background, that's not surprising. But
right. So if you're thinking in that umbrella of concepts as scientists, you're trying to interact with this cannabis community, You're trying to understand legacy and the culture and fads, and I had to understand what customers want,
Right.
but I've got to give them what they want and need at the same time.
And how they can process it. Like, they have a certain language and information structure that you kind of have to fit things in.
You back into the plants, and you're like, even if I have 20 Girl Scout cookie plants here, every nug is going to be subtly different, and every growth is going to be subtly different. Every time I grow it, it's going to be subtly different. The media, the nutrients, how hot it was, how much humidity. So each one of those is going to change, and then they have this fancy word now, chemovar, which I won't accept, which is just a layman's term for not renaming something in cannabis, because we've got a cultivar and we had a chemotype, and those fit anything I need to discuss with this perfectly.
Well, yeah, I mean, that actually, I can comment specifically on that. So I interviewed someone that his mentor was the person that coined the chemovar term, and he pointed out that term was never supposed to be used the way it is actually being used now. He said it was supposed to be a synonym for chemotype at a time when chemotype was not a common term that people were using.
Sure. Why would you do that? So it's just been picked up as this is we're going to co-opt cannabis term,
exactly,
right? Like strain.
Yeah, exactly,
we want to call it cultivar, and I get caught in between.
Yeah.
But chemovar confuses what is happening in my mind. It relevates cultivar and chemotype. So it's like I can give cultivar is extremely important because it's the genes that do it, but nature versus nurture. Anytime you bring nature into it, you're changing the chemotype into
especially
something
with the terpenes.
Subtly different.
Yeah.
So each batch at that time and space in the amount of it is a chemotype, and I process it and make this is a chemotype, and then once that's there, it's gone.
Right. 6 weeks from now, the chemotype may be different.
Yeah, genetically it may drift. I may add new nutrients. That law of soil that I got might be pH different. So chemotype and cultivar are importantly separated and not mashed together into chemovar, which is trying to track down the explanation, which is always what I like to do for full spectrum, which is the plant's chemical makeup. This doesn't describe cultivar or chemotype. And with full spectrum, we ask, what does that mean?
What's the spectrum?
They say, it was lab tested. That's what we've gotten back. Sorry, I got a name drop, but it is a company in Oregon who may or may not sell CO2 cartridges. Just told us it had been lab tested, and that's why they called it full spectrum. And it's very shocking to think that somebody could be like, I give it to the lab, and I go, did the lab tell you that?
Right,
because I'm trying to go down the chain, like the lab goes, the customer asked, is this full spectrum? And the lab goes, yeah, it's full spectrum. And now I got a customer who's an extra company putting it on their box and people buying it. Thinking it's all, well, how did the lab come up with this idea? And the labs, you go back to the lab and you say, why did you tell them it was full spectrum? And they tell you, oh, it had multiple cannabinoids in it. And okay, well, doesn't everything have multiple cannabinoids in it? Even the most purest distillate has multiple cannabinoids in it. So I apologize to you.
That's exactly my point. Even like an isolated CBD product, 95% pure. Well, there's still other stuff in there. So is it full spectrum?
CBD might not be, but everything else, like distillate or isolate, is aptly called isolate because it could be 99.98 as a crystal.
Right.
But like a distillate is as a syrup.
Yeah, I mean it's like a semi-solid. It's like I relate it when for people that aren't familiar with distillate, I describe it as sort of like hot glue, like slightly cooled hot glue.
Yeah.
Because it is it's like a like a very 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 thick syrup and and I mean and that's not even a place for labs to even comment. Like labs are in the business of realizing data. That's what they should do, and they should do nothing else other than that. Making sure that the process of realizing data is solid and backed by good quality controls and everything. And then how people interpret that data or make claims about that data, that is for other companies to do. And I do get disappointed when I see cannabis testing labs trying to get into like the marketing game, trying to influence
like
marketing of the industry. That's totally out of place. And
like telling you what CBD is good for, or what terpene.
Oh my gosh,
good,
I really hate those charts that you see in dispensaries that have the grid, and it will say have all the
inflammation,
and all the dots and everything, and it's like, okay.
The biggest one is limiting antidepressant. Right. I'm like, so when's the last time you gone to the doctor? And you're like, I'm feeling blue, and I'd be like, have some oranges,
right?
Or lemons, squirt some lemon, like.
Well, and it gets into, something you probably have appreciation for, especially given some of the research that you've done for your graduate studies. But, something that's really rampant in the cannabis industry, but it's also really rampant in the natural products industry broadly, is people wanting to look at like cell culture models or really simple in vivo tests and then extrapolating pharmacological data that supposedly is applicable to a human being, and then marketing that
antifungal or
just all these different, So, for instance, one common thing that I talk about, and it's evolved since I first was teaching on this, but when people were talking about CBD and CBC being these really awesome antidepressants, that was not based on human clinical data. That was based on mouse studies that were doing forced swim tests and tail suspension tests, which if anyone listening that doesn't know what a forced swim test or a tail suspension test is, this might disturb you. But anyway, so a forced swim test is when you train a rodent to swim in a pool of water and find a platform. And then eventually you take away the platform and you see how the rodent, basically how long will the rodent try to find the platform before they drown. That's one study. And so they'll give these rodents different compounds to try to see how it affects their behavior. And basically they use that behavior of how long are they willing to fight to survive, essentially, to relate to antidepressant activity or anti-anxiety activity. The tail suspension test is where, and this one really might bother some folks, but I think you need to know it. Rodents are literally suspended from like a wooden plank above a pool of water, and you measure how long they fight to survive before they give up and just lay there, just suspended. Yeah, and the same thing, they'll give them compounds and extrapolate that behavior to relating to antidepressant, antipsychotic, even anti-anxiety effects in humans. So I think if people understood that more, they might be a little less willing to jump into some of that marketing material that says, oh, this is 10 times, the antidepressant effect of this. Well, how is that medical claim derived? And if it was a tail suspension test or forced swim test or something like that, while that may indicate something interesting, there's already a huge issue in clinical research philosophy right now about whether these types of studies are even meaningful at all.
So, where do we get meaning then? Two scientists in a room again, and I say, how do I bring meaning to customers who want meaning? And I need to know more about them.
Yeah.
And this is, again, the dangerous side of having people share too much information and allow them to share too much information. Think you have too much of their information. But this is based on the human genome work that I've done, and they wanted to encourage everybody to get genome tested. Everybody go to your doctor and pay for your $30 test, because we got to start figuring out if you have all these kind of diseases they don't have really mapped out, and it's only then. So everybody started cutting off their breasts. And they had to roll back because it was like a 56% chance if you had these two BRCA genes that you may develop breast cancer. And it wasn't like 100% chance, but Angelina Jolie goes out and gets a mastectomy. And so they kind of had to roll that back a little bit. But in that process, we needed to know more. Just genome is not enough. Well, I need to know what happened to your parents, who your parents are, what symptoms you're having, what your outcomes are, what your intolerances are, if you have allergies. So when I tell you I'm going to make a product for you, when I tell you I need you to communicate back to me if it helps you with being anxious, I need to know what kind of anxious we're talking about. And so you have to develop all these rules so they can easily, like easy text, Instagram, social media, go through their history and feel comfortable that you're safely preserving it, so that you can get at the meat of telling these class of people who are anxious, that CBDC might work for them,
Yeah.
but these class of people are anxious. That's actually just a version of depression or some other thing that where you want to stay away from CBC or, like CBD and THC and might help you, but still we're unsure. So you get that from feedback from people, but it has to have this context.
Yeah, it's got to have some control. And you're touching in on another big aspect, discussions that are going on right now in psychology research, and like how do you measure depression, anxiety, just overall dissatisfaction with life outcomes, etc. And it wasn't until the past several decades that people realized, like, oh, maybe like a 0 to 10 scale for pain is maybe not good enough, because people have different types of pain, and also, when you deal with psychological conditions, there's a huge amount of subjectivity to it, as far as, my pain and your pain are very different experiences. And even finding some objective measure of some sort of signaling that's going on in the brain is not going to tell you how your pain compares to my pain as far as how we feel it. The same goes for things like depression and anxiety, sadness, malaise. And so when someone makes a claim, for instance, to bring it more directly to like CBD and the epilepsy research, you can't just say CBD is good for epilepsy. Epilepsy is a broad term that
Parkinson's.
There are all sorts of different forms of epilepsy, and medical research hasn't even teased out those nuances yet. Like, we're at the beginning of understanding that there's a lot of different forms of epilepsy caused by different things with different mechanisms that present similar symptoms. And we have to understand that when we come up with a term for a disease condition, that term is usually representative of a group of symptoms, but those group of symptoms can have varying causes.
But even as you say that, to me, I'm thinking epilepsy is a type of seizure, an event of muscle and rollback and anything like that. So we're speaking different semantics because we're on that communication. But to you, it sounded like epilepsy is one specific thing. But I'm thinking there's still other seizure events, and I just didn't know they're all called epilepsy because we're in different semantic ranges. But I wasn't naive to think when I say lemon remedy helps with epilepsy that it's a specific type of epilepsy. Or it's a specific type of seizure. It's not grand mal or other types of seizing or seizures or something like that.
Well, and that's where the communication piece is so important. It's like if something is helping someone's epilepsy, it's like, oh, well, what form do they have, or even just like what's their age and what are their seizures typically like? What's the typical frequency, and how's it affecting those things?
Let's wrap it in a bow because of what you talked about in the future of cannabis.
Yeah.
It's like, what am I searching for? The meaning of life, the meaning of cannabis. It's like, why? What is this endocannabinoid system? Why is it so important? Why isn't there a single medication that addresses endocannabinoid deficiency? What are phytocannabinoids? Why is there something that exists in nature? Is it a nutrient like a starch or a protein or a vitamin that we need? It's given to babies the moment of birth from the mother, and these are the most important things infant needs to survive, or what comes out of the breast milk.
Just to have the sensation of needing to feed.
And a pharmacologist was explaining to me phytocannabinoids are processed in the liver, and endocannabinoids are more brain type molecules, are isolated or metabolized. And so I'm thinking, because I was thinking maybe this is a vitamin that humans and mammals need, and because of this THC bastardization, that we've moved away from this vitamin that society needs. And I was maybe being too romantic about it and too over, like, we definitely need all cannabinoids in our system, everybody, right? And then I was kind of backtracking with the way phytocannabinoids are metabolized and talking with somebody, and he said, well, it's just really exercise is really what pushes the endocannabinoid system moving forward. And you get the runner's high, and that's where you're actually producing all the endocannabinoids. And I thought to myself, like, youth and exercise and movement and endocannabinoids is what the meaning of life and health is. And as a society, we've been sedentary and we moved away from that lifestyles that are activity every day and sun cycles and noon cycles and having this regulation of our sleeping and our digestive system. As we become sedentary and look at screens and have this lifestyle, we need more phytocannabinoids in our society to deal with leaving behind that running and catching our own food and exercising as a lifestyle and living for only 30 years. We need, as a society, all this proliferation of phytocannabinoids to plug in into that.
Several other people I've talked to have sort of mimicked that message, that one of the best things you can do to support your endocannabinoid system, and it's the message that we've all grown up with: exercise more and eat healthier. And the eat healthier thing, I think, is a very interesting piece, because we're learning that a lot of plant compounds interact with the endocannabinoid system, and our foods over time have become less and less rich in a lot of these phytonutrients that would do that, including things like flavonoids and certain terpenes and things like that. And one example I use is corn, given how ubiquitous it is in our diet, that at one point corn was bred for a high oil content, and one of the big terpene constituents of that oil was beta-caryophyllene, and beta-caryophyllene interacts with CB2 receptors and possibly other components of the endocannabinoid system. And that was some dietary interaction with the ECS that we were having early on. And then between I think it was like the 40s and the 60s, in that time, we started to breed out those oils, because at the time we didn't have a way to value them. It just wasn't really in our consciousness that was something we needed to care about. So as a food product, corn started to become less and less abundant in beta-caryophyllene and other compounds. I'm sure a similar story is true for a lot of foods. And what's interesting about the corn piece is now researchers have learned that having those terpene-rich oils actually helps reduce the need for pesticides of corn. So now they're starting to breed it back in. And so it may be interesting to see, with the combination of exercise and finding phytonutrient-rich foods, and regardless of the fact that we don't know exactly what's going on, but just knowing that they are interacting with the ECS, and that it's generally recognized now by the folks that are studying the ECS the most, that the endocannabinoid system is essentially a foundational physiological system. It's connected to everything else, and it's driving a lot of homeostatic processes. And I get a little anxious talking about homeostasis, because I think it's a concept that people often mischaracterize, but it has something to do with that overall balance of the body. And so absolutely, we used to be more active, our foods used to be more phytonutrient dense, and we move around a lot less. Our foods are a lot blander, more bland compared to what they used to be, and so that's perhaps where this all fits in. Is if there is a deficiency, that would explain a lot of that, and being able to stimulate that system. And
I say the proof is in the pudding. I mean, over the past 40 years, we've gotten better with air quality and water quality as a nation, right? And less cigarettes. But I mean, you look at movie stars from the 80s, they look like old baseball gloves, and they're all like 40 years old. And you look at all people in the cannabis industry, and they're all like a bunch of babies, soft skin, nice teeth, and, like pretty decent overall kind of health, because they've used cannabis all their lives, or 20 years. And so I just look at that as one fundamental, empirical piece of evidence.
Yeah, and there's interesting research showing that like THC stimulates metabolism. It changes the way glucose is managed in the body. And I interviewed Dr. Ethan Russo, and he was telling me about some newer research being done on the gut microbiome and cannabinoids, and showing that THC tends to promote a healthier balance of microflora in the gut. And obviously, it's now generally well known that the gut microbiome, is a critical piece of the immune system, as well as the nervous system and everything. And
well, that's with Crohn's, and as you know, that's very good help with Crohn's. I've had my family has history with digestive diverticulitis and stuff. So I know the pain and pressure people go through with Crohn's disease, and cannabis medically, the papers have come out.
Yeah.
The verdict is in.
Yeah.
It helps and could cure people with Crohn's disease. And I mean, when you're drinking Ensure for your life, you want to get back into eating and interacting with real foods. Yeah, cannabis can help, those people. And I like the applications of cannabis and medical research that's real concrete, like epilepsy and children's seizures being cured, and movement-based Parkinson's being lessened, and Crohn's. And if you put RSO on certain types of topical skin cancers, they can vanish.
Yeah.
So those practical solutions are unique to the individual. It might not work for everybody, but making products that people can try.
Yeah.
See if they avail these symptoms. That's what we're all about. So.
Yeah, I love it. Well, I'm going to go ahead and get this wrapped up. I think we've been going for at least an hour and a half or more, if not an hour and 45 minutes. I want to give you a chance, if there's anything that we haven't gotten into that you wanted to spend a little time talking about, we can do that now. Otherwise, if you just want to tell our listeners how to learn more about your company, the work you're doing, how to keep track of your development and all of that, I'll just kind of give you the platform, whatever you want to go into.
Yeah, no, so all of our media is online. We're got a pretty good presence online, so you can reach out to us on social media or extractioneering.com. And then there's some talk about our research interests. Of course, we want to get really expensive machines in this industry.
Of course, we all do.
Test all these products and look into what these compounds are. But more or less, what I really would like to promote, and we're trying to figure out what we are as a company and our ethos. And of course, we've got a lot of selections to choose from. But I honestly really feel like THC needs some help.
Yeah.
And we got to start pushing back against the whole intoxicant premise. And it's a real babe in the woods moment. We don't want to rush out there and be like, we're a bunch of drunks and you have to accept us for it or something. But we had to break this chain.
Well, I think
at some point, and because THC is what people are looking for in most of these preparations.
Yeah, and you're definitely not the first person to mention that. I mean, several of the healthcare professionals I've interviewed have said the same thing. CBD is great, but THC is really where we're seeing a lot of our improvement. And I think maybe, I think harm reduction education goes a long way for that, helping to explain to people that there's a way you can engage THC to avoid having an uncomfortable psychological experience, and that sort of thing. And that can, I think, help.
That goes along with each person has a different and unique endocannabinoid system, right? So I that's where going to be our message really, is to be back and say, THC, it's for you and me. It's not the villain.
Yeah.
It's, as they try to choke the hemp down and choke everything out to try to just make a point, right?
Right, yeah.
Just trying to stand on that last island.
Right, as the water's rising.
As it rises and say, no, you can't have this because we know it's the most important thing or what you want.
Yeah.
We're getting lost in the weeds there. Stuff's too important. So it's gonna have to be just push it into their spot and be like, this is not a problem.
Well, and I was talking to a nurse about this, and a comment she made that that stuck with me. She said that particularly for people with medical issues, that THC can really help. She said a lot of those folks, if they truly have a strong deficiency where that's really satisfying that, she said, one, those types of people tend to develop a tolerance really, really quickly to any sort of uncomfortable effects of THC. And a lot of times they tend to not get as high as a lot of people do because their bodies are getting something that it kind of needs, and it's sort of moving them closer to a baseline rather than pushing them far above the baseline. And I think that's something important for people to realize, is going back to this, that everyone interacts differently.
The plateau is an explanation.
Yeah.
Somebody who doesn't have it often, it spikes like this. It comes way up, like a GC, and it takes a while to come down, like 3 hours or something. And so where I am at is, so I've created this line, and so I move like one tick above it and come back down to that line. And as long as I interact with it, I am on that plateau, and only when it's been a while I start to come down. So what's been a while is I don't use THC or cannabis while I'm sick with a cold or a virus, because I feel as though it has some this depressant of the immune system in the same way it could help me with inflammation or allergies to kind of slow that stuff down. Is the same thing where it kind of slows my immune response and what it's attacking at that particular moment. Not immunity, me fighting off things every day, just when the immune system needs to be on high, it doesn't need to be slowed down.
Exactly. No, that's
So when I go off of it for 4, 5, 6 days because of nasal congestion or whatever, and I come back, I spike right up there again, and it's very, very uncomfortable.
Yeah.
But 2, 3, 4, 5 circumstances later, I'm on the plateau.
Yeah.
And only the weirdest of circumstances.
Yeah.
And so with THC, the temperature at which you use it, the percentage of THCA to THC, which we will be transparent and report on, recommendation about what the voltage is in for pens, and then of course you mentioned edible versus vaporization, but then this product at the same temperature and amount, and this product in the same temperature amount is going to be a completely different experience.
Yeah, and that's something, I mean, I don't know if it's something we need to spend time or not on, if you want to, but it's something that I actually thought about and then didn't think to ask you. But about the temperatures, is there anything you want to specifically comment on regarding how folks are using these different adjustable devices? And
so we want to recommend a device and a temperature when it comes time, but each device and setup is unique to the device and the setup. And so usually you can measure the temperatures. We're usually on glass at 440 degrees. Which is any one of these products to vaporize. But if you have more need for cannabinoids, you're going to be inching that up.
Yeah.
And going too high would create a problem. But you would be uncomfortable at the level at which you'd be using it, and it would taste poor. Both in cartridge. So if you have, I guess I can circle back. So none of my products taste poor. Maybe if you're a product company, you can't separate what tastes poor and what's burning and what tastes poor. It's just fine. It's vaporizing. But one of these products tastes how I want it to taste, which is good. And if it's too hot or it's too high and it tastes poor, it's too hot and it's too high. It's not because my product is tasting bad. So
and then you're affecting, like particularly in a vape pen, if you're burning something too hot, you're not only affecting that draw that you're taking, but you're affecting the material that's sitting in that cartridge for future draws.
Yes, and so then just to wrap, with the wax that polymerizes that's left in here, when you put this liquid in a vape pen, it gets wicked up and turned into vapor without collecting. When the product still has a wax, forms a shellac on the vape pen. Almost 2 or 3 uses. Yeah, that's great. The first couple. Then it forms like this wax membrane on the thing itself, and so it gets poor performance in the cartridge down the line. And so it's another reason why we make sure everything has been taken out in terms of wax.
Is that why some vape pens, after a while, they get almost impossible to draw from?
That is 2 things. Also, the liquid gets into the draw tube. And that's what the preheat function is for, is to loosen up the plug of liquid. It's a little bit more viscous of a product. These products don't typically have those problems because they remain, even though this is a room temperature product now, I took it out of 90 degrees, but they remain fluid because they're separate molecules, because they're extracted in the right chemistry. So it works with all of the vaporization intent down the line where other products don't.
Well, and that relates to my question earlier about additives and everything, and talking about solvents that a lot of people don't realize, like terpenes are solvents, some cannabinoids are solvents.
So your body makes acetone. It is a solvent. You drink alcohol at 40, 80%. You have no problem with it. You make lactic acid and alcohol as a solvent. If I steam distill terpenes off of plants, I get something called a plant hydrocarbon.
Yeah.
Which is what dinosaurs and plants is what hydrocarbons are made out of, right?
Yeah,
so I steam something and I'm getting a hydrocarbon in my steam distilled terpenes because it's a natural plant process.
Right.
It's a short chain carbon with the hydrogens on it. And it's at 10% in steam distilled preparations.
Yeah.
So it's like the concept of what is a solvent, when is it dirty? It's toxicological threshold.
Yeah.
We use clean solvent. So it's important to us to get our solvent from the distillery. So they test it as it comes out and it's put into our tank. Yeah. The only impurities are the other propane and butane. So the impurity in propane is butane and the impurity in our protein is butane. And basically it's 100%. They take the tanks and they clean the insides out and they restem all of them because LPG tanks are notoriously in a dirty, they don't worry about that. So they give us a certificate, they give us a stamp on the tank, they tell us the documentation of how it was transported. And all of that is going to be, our tank number is going to be listed on this product number so that we know where that came from. Yeah. That's
really good to hear because, and I promise we'll finish this up, but you keep making me think of other things that I want to mention. But going back to GMP and that sort of thing, something that is less enforced in the food and dietary supplement world is definitely enforced in pharma. And it should be enforced anytime there's extraction going on. But you were talking about why not to recycle solvents in a closed loop system and everything. Well, one reason why that is oftentimes really difficult or impossible to do in a pharma setting is because you have to validate the purity of the recovered solvent, and it has to be a certain purity before it can be used again in the process.
So imagine government saying, "Oh, that stuff's dangerous. You got to reuse it every time until it's done." I'm in the business of making the products. You're not in the business of making them. So I want, a decent product. But like I said, all the acetone, I told you what we use for steel and PTFE and glass and everything. The acetone that we use to clean it, it's HPLC grade to the top.
99.9 plus
percent. It's not the stuff from the hardware store, Greasy Joe.
You're not using naphtha?
No. So it's all in it. It's just $20 more.
Right.
It's not a big deal. But it's a big deal to us because we know that every time we use our acetone or ISO, it's of that level of quality. It doesn't have any of those other impurities, and it's in like a container it's supposed to be in.
Yeah. Well, I hope that everyone listening to this, by hearing us talk about some of these nuances, maybe it'll spur some new questions. I'll start asking producers.
And then you say, how do customers know? You tell them to look at this and ask all those people these questions that they learned here today and try to understand more about who they're getting their products from.
Yeah, and particularly dispensaries that are buying, things in bulk and reselling. I mean, this is something like I strongly recommend devise a simple quality questionnaire to issue to vendors. And if they have a hard time answering questions about chemical compatibility or the quality of inputs and that sort of thing, like that's a huge red flag. And there are enough producers out there, you can hunt down the good ones that are trying to do things right, that are measuring their own quality and managing that you don't have to push out bad product just for the sake of having something on the shelf. And hopefully that'll continue. But
cool.
Cool. Well, thanks so much for being willing to give me so much of your time and chat.
No
I've really enjoyed it. It's been great. And like I said, as things progress, if you ever want to do another drop-in, or if something pops in your mind, some topic that we want to do a deep dive on, just you've got my contact now. Hit me up, and we'll do this again.
Good enough. Thank you.
Cool.
Thanks
Awesome. Well, take it easy and
yay.
Yeah. And as I say, stay curious. Everyone listening, if you want to find out any more about Extractioneering, check them out on Instagram at Extractioneering and extractioneering.com. And then for Curious About Cannabis, you can find us at Curious About Cannabis or cacpodcast.com and connect with us on YouTube, Instagram, Facebook, or Twitter. Thanks so much for listening or watching, and I'll talk to you again very soon.